Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the RH grouping and also we studied about the erythroblastosis fetalis, that is also called as hemolytic disorder of newborn. So up to that we have studied, and now we are talking about how exactly the blood clotting will occur. The blood clotting is also called as blood coagulation, and how exactly this mechanism is happening, so that we are studying here. Before starting this particular one, I am telling you very clearly, complete mechanism was not mentioned in NCERT. Please remember, in the complete mechanism, you will need to study about all the factors. All the factors we need to study, and there are two different pathways here. Intrinsic pathway and extrinsic pathway are there. And these things were not given in NCERT. So please remember, in the NCERT, only the last two steps of this particular clotting mechanism was given. Actually, it is a cascade of mechanism. Please remember, when we are using the word called cascade, so if there are some sequence of the steps, that is called as cascade. So in this way, here we will, I will show you the diagram, but please remember, we are not studying all the steps. So in the examination, if they are asking about the information that is not there in NCERT, so you can simply leave it. But anyhow, please remember, they will not ask the questions on information that is not there in NCERT. So please remember this, because the pathway is not that much easy. So to remember this one itself, it will take so much of your memory. So at this particular level, you don't need to worry about this. Only the last two reactions, since they mentioned in NCERT, you should compulsorily remember those two reactions. Clear? And there is one more sentence that is linked with class 12th chapter, that is microbes in human welfare. So that also I will tell you. So these points, whatever they mentioned in the NCERT, you need to compulsorily remember. Remaining information is just for understanding purpose only. That's it. You don't need to remember that particular information. Clear? Now let us start with this. And what is this blood clotting? So where it will clot and when it will clot? See, when blood is actually flowing in the blood vessels, there it should not clot. There it should not clot. So please remember, already we studied, there is a chemical that is called as heparin. Heparin. Who will produce this one? Already we studied this. Who will produce this heparin? Basophils. Yes or no? So basophils are releasing this heparin, and this heparin is called as anticoagulant. Why it is called as anticoagulant? Because it is preventing the clotting of blood when it is flowing in the blood vessels. Yes or no? So in this way, it is helping in keeping this fluid, keeping the blood in liquid condition. Yes or no? So in this way it is helping. That's why it is called as, heparin is called as anticoagulant. Anyhow, that point, once again at the end of this lecture, I will cover that particular point. And now we are focusing about clotting of the blood. So suppose in general, when we are getting some small cuts, so what is the average time? What is the average time, or what is the average duration? So for this blood clotting. So please remember here. So here generally information is same only, but in the examination they may ask in two different ways. So what is the average time for the blood clotting? So generally we say that it is 8 minutes. 8 minutes you are saying. So this is the average time, but in some books they are giving this as 18 to 15 minutes. But whatever it is, within this time clotting will occur. Within this time clotting should occur. So since clotting is occurring within 8 to 15 minutes, now generally what they are saying, between 1 to 7 minutes. 1 to 7 minutes, generally they are calling this one as bleeding time. Bleeding time. Please remember, information is same only, but the way in which they are asking the question is very, very important. So before 8 minutes, there is a chance of bleeding, but after 8 minutes, there is a chance of clotting. So in this way, generally standard information, if you are observing, 8 minutes, a minimum of 8 minutes it is taking for the blood clotting. So that means before to this will be the bleeding time. There actually the bleeding is occurring. So in this way, same information they can ask in two different ways. So please read the words very carefully, and accordingly you please select the answer. Clear? And minimum we are saying that 8 minutes time it is taking. Nice, very good. And how exactly this clotting is occurring? This is the next important point. And please remember, now I will show you a diagram. So please try to understand what is the meaning of the words first. Suppose this is a blood vessel. Suppose this is a blood vessel. Okay. Please remember here, damage to the blood vessel. So damage of the blood vessel will result in the blood leakage, yes or no? And now this should undergo the clotting. Now please remember, sometimes the damage may occur from inside. That means this blood vessel wall of the blood vessel is having some thickness, right? It is having some thickness. And here the damage of this particular blood vessel sometimes may happen from inside. That means from the inside of the blood vessel. Inside means, suppose this is the blood vessel. For the blood vessel anyhow, at the end of this particular chapter we will study. So it is having some layers. Yes or no? Outermost layer is called as tunica externa. Middle is called as tunica media. Innermost is called as tunica intima. In this way, 3 layers are there, and actually this tunica intima layer is, innermost layer is generally having squamous epithelial cells. Endothelium generally it is having. Yes or no? Now what I am saying here, if the damage is happening from inner side, that means first inner side wall is damaged. This is called internal damage. Suppose due to some other factors, if the damage is happening from the external side, also damage can come here, and sometimes damage can happen from inside to outside. So in this way, from which location exactly the damage is taking place, based on this, 2 pathways are present. Anyhow blood clotting will occur, but who will initiate the blood clotting? So based on this, if the damage is happening from the inner side, now intrinsic pathway will occur. If the damage to the blood vessel is happening from the outer, beginning is from the outside, now this is extrinsic pathway. So in this way, 2 pathways are present. One is intrinsic pathway and the other one is called as extrinsic pathway. So in this way, intrinsic pathway, extrinsic pathway. But whatever is the pathway, ultimately when they are coming to this, from here onwards, all the steps are same. All the steps are same, but initially. So if the damage is from the internal side, some factors are involving initially. If the damage is from extrinsic side, some other factors are involved. But whatever it is, all these are ultimately involving in these common steps. From here onwards, whether it is intrinsic or extrinsic, whatever it is, steps are same only. And here if you are observing, here they have given some numbers here, Roman numbers they have given. So with the help of some Roman numbers they are mentioning all these, but originally they are having a fixed names. And here, even though number wise if you are observing, here we are writing up to the 13 number. Even though you are writing 13, but originally how many factors are present? Only 12 are present. Why only 12 are present? Because number 6 is not assigned to anything. Number 6 was not given to any clotting factor. There are no clotting factor with the number 6. So since number 6 is not there, even though you are giving up to 13, so total count if you are doing, so it will be minus 1. 13 minus 1. So 13 minus 1 means 12 clotting factors only we are studying. Only 12 clotting factors are present. Okay. And their numbers are clotting factor 1, 2, 3, 4, 5. Please remember there is no 6. So directly go to 7. 7, 8, 9, 10, 11, 12, 13. In this way they have given the numbers. Clear? And now I will simply read the list of these, and you don't need to remember the complete list. I will tell you what to remember. Okay. So first let us go through the complete list of these clotting factors. 12 clotting factors are present. Factor 1 is fibrinogen. Whenever, already I told you about this. If the word pro is present before the word, if the word gen is present at the end of the word, this pro and this gen. So these words represent that they are in inactive form. Please remember all these clotting factors, they are present in the blood, but these factors are normally present in inactive form. Only when there is a requirement for clotting, blood clotting, only then they are activated. Until then they are present in inactive form only. All these are normally present in inactive form. So if there is a blood clotting requirement, then they should be activated. Please remember this very important word. Okay. So factor 1 is the fibrinogen, factor 2 is the prothrombin. And this whatever I kept in the yellow, please try to remember this. Anyhow, regarding fibrinogen, prothrombin, even reaction also we will study. In the NCERT very clearly they mentioned about the activation of these. So these two are compulsory. You need to compulsorily remember this. Fibrinogen is factor 1, prothrombin is factor 2. This is compulsory. You need to remember this. Factor 3 is tissue thromboplastin. This is called the tissue factor. Okay, nice. But you don't need to remember these names. But factor 4 you should remember this, because this calcium is ion. Remaining all these are proteins. Except this calcium, remaining all are proteins. Only calcium is a non-protein. It is a mineral that is acting as clotting factor. and its number is factor 4. Please try to remember this. Okay, very important for examination. Next one, factor 5 is labile factor. It is also called as a proaxelerin. You don't need to remember. Factor 7 is stable factor or proconvertin. You don't need to remember this. Factor 8, 9 and 11. 8, 9 and 11 I kept in yellow. You don't need to remember these names. Actually, you don't need to remember the names, but at least remember the numbers 8, 9 and 11, because these three are these actually in general loss or absence of any of these factors will cause a disease called as hemophilia. At the end of all these things I will tell you. And in that hemophilia also you are talking about hemophilia A, B, C. These three are very important because in the principles of inheritance and variation, that is class 12th chapter, there you will study about this. In the NCERT main textbook, they have not directly talked about hemophilia A, B, C, but in the QR code content they have given about hemophilia A, B and C. Since they have given in the QR code, that is also part of NCERT, so you should know about this. So one sentence I will tell you about 8, 9 and 11. So please remember this. So what is this 8? Anti-hemophilic factor. Hemophilia is the disease. It is the anti-hemophilic factor. So it is preventing the hemophilia. That anyhow I will tell you separately. Factor 9 is plasma thromboplastin component. It is also called as Christmas factor, but you don't need to remember the name, but remember factor 9 is related with hemophilia. And factor 10, Stewart-Prover factor. No need to remember, leave it. Factor 11, plasma thromboplastin antecedent. Name is not required, but simply number, try to remember this. Next one, number 12. Number 12 is Hagman factor. And number 13 is fibrin stabilizing factor. So these are the total 12 factors. Numbers are given like this, and number 6 is not present. Clear? And out of this, what are compulsory with number and name? One is important, second is important, third is important. That's it. These three, numbers and names, you need to remember. For 8, 9 and 11, simply numbers you just remember. Absence of 8, hemophilia A. Absence of 9, hemophilia B. Absence of 11, hemophilia C. Clear? So simply at least numbers you please remember. And these are the important factors here. In this way, 12 blood clotting factors are present. Out of this, one and only non-protein mineral that is acting as blood clotting factor is calcium, and its number is factor 4. Please remember this. These are fixed numbers. You need to remember like that only. And now we are talking about the mechanism. If there is a damage to the blood vessel, maybe from inner side, maybe from outer side. If the damage is from the inner side, this is the pathway that is happening. 12 is activated first. This activated 12 will activate 11. Now 11 is activated, and then now this activated 11 will activate 9, number 9. And now this activated 9 will activate the 10. So the 10 is activated. This is the intrinsic pathway. In the extrinsic pathway, what happens? Now here tissue factor is coming here. So number 7, this is inactive. Now it is activated. Now this activated number 7, now this is activating this 9 into 9 activated. 9 activated will convert 10 inactive 10 into 10 activated. So in this way 10 activated is there. Now from here onwards, whether it is intrinsic or extrinsic, now these are the common steps. So now let us see what is happening here. Now this number 10, activated number 10, that is helping in, activated number 10 is helping in conversion of prothrombin. Pro is inactive. So prothrombin into thrombin. What is prothrombin here? It is number 2. Yes or no? It is converting number 2 inactivated into activated number 2. That is the thrombin. So prothrombin is converted into thrombin. And here we are talking about activated 10A and also activated 5, fifth factor activated, and also this is the PL, platelet phospholipids, and also calcium ion. And here in every reaction you just see this. Every reaction you just see here. Calcium ion is compulsory. Without the calcium ion, this blood clotting mechanism is not there. So in this way, sometimes what we will do to prevent the clotting. If you want to stop the clotting, the simple logic is stop the action of calcium. So you just simply make this calcium unavailable. How you can make the calcium unavailable? Very simple. You just add some chemicals. There are some chemicals like EDTA. I will tell you once again at the end. There are some chemicals like EDTA. They will go and bind with calcium. So this free calcium will be bound with EDTA. so that there is no free calcium in for the reaction. If there is no free calcium for the reaction, blood will not clot. In this way, you can stop the clotting of blood. So that's why generally what we will do. Suppose you are collecting some small amount of blood for some tests, generally at the lab level. So for economically, if you are thinking, so what we will do? We will take a tube. In the tube, we will add EDTA. Collect the blood and then you just add blood to it and mix properly. Now EDTA will immediately bind with calcium ions, and calcium ions are now attached with EDTA. Now they are not available for the reaction. If calcium is not present, what happens? Whatever the blood you have kept in the tube, now it cannot undergo coagulation. It will not clot. It will be in the liquid form only. Please understand this. So in this way, calcium is very, very important, and not only EDTA, some chemicals are there. Once again, I will tell you at the end. Clear? And in this way, calcium is very important. Calcium ions are very, very important. So finally, what is the important information they have given in NCERT? Prothrombin is converted into thrombin. So prothrombin is inactive, thrombin is active. And how it is converted here? It is converted by thrombokinase or prothrombinase. Prothrombin will be converted into thrombin. You can also call prothrombinase as thrombokinase. Both the names are same only. And what is this one? This is the group. And this group is that thrombokinase or prothrombinase, so that it is converting prothrombin into active thrombin. And now this activated, once thrombin is formed, now what happens? This thrombin will convert fibrinogen into fibrin monomers. Fibrinogen, gen inactive into active. What is the active form? Fibrin. In this way, fibrin monomers are formed. And these fibrin monomers, they will cross link with each other and they will form a mesh or network like structure. They are forming network like structure. And in this network, now there are some small gaps are there. And these gaps are filled with the blood cells. Blood cells will come and they will attach to this. In this way, they are completely blocking that particular place. So in this way, whatever the damage that has happened to the blood vessel, there is a damage happened here, right? Now here what is happening? Fibrin is making this mesh, and to this mesh what are happening? Blood cells are coming and they are attaching to this and they are completely closing this particular damaged portion. So once it is completely closed, what happens? Now blood clot is completed. Blood clot is completed. Now there is no further loss of blood. In this way, loss of blood, you can simply call it as bleeding. So bleeding can be stopped. Understood or not? But in this process, please remember, since blood is coming here, so cells are trapped here. Suppose there is a mesh-like structure here, cells are trapped here. So some fluid can go out. The fluid, a small amount of fluid can go here, and this you are calling it as serum. Yes or no? That is the reason why in the previous lectures also we studied what is serum. Serum is equal to plasma minus clotting factors. Plasma minus clotting factors. This is why you are saying serum as plasma without clotting factors. This is what. So in this way, please remember what is the ultimate and important point here. It is the blood clot. This blood clot is mainly having what? Fibrins, fibrin proteins, and this fibrin proteins. Now these clot is again stabilized by there is another factor that is called as number 13. Yes or no? So it is the fibrin stabilizing factor, right? So in this way, initially it is in inactive form, and this thrombin is helping in activation of this. So it will finally help in the stabilization of this fibrin clot. So in this way, blood clot is mainly what? Fibrin protein clot. Blood clot is actually fibrin protein clot. So now anyhow some cells are attached, blood cells are attached, and they are completely blocking this. In this way, now that whatever the damage is happened, now the damage got sealed. Now it got sealed. Now there is no further loss of blood. So in this way, clotting of the blood is very, very important at that particular location. Clear? So in this way, this entire mechanism is happening. Now in the NCERT, what they said is, they are saying in this way, blood clot is actually having the fibrins. And how these fibrins are produced here? This fibrin is produced from fibrinogen, which is an inactive protein present in the blood. And how this fibrinogen is converted into fibrin? By the thrombin. And how this thrombin is produced? It is produced from prothrombin. And for this one, prothrombin is inactive, thrombin is active. And this inactive prothrombin is converted into thrombin by thrombokinase or prothrombinase. And that is actually having these components. And how these components are obtained? In these mechanisms. So regarding these mechanisms, they have not mentioned in NCERT. Only from this one only they are talking in the NCERT. So please remember this. So this is how they are simply, in simple, they are giving only this much. Prothrombin is converted into thrombin by thrombokinase or prothrombinase. Now this activated thrombin, now it is helping in conversion of fibrinogen into fibrin. So now this this is the fibrin monomer, this is the functional one, this is the functional one. And now these fibrin monomers, they are interlinking with each other and they are forming a mesh-like cross network. And then finally they are helping this particular one. That's why clot or coagulum is network of fibrins. Very, very important point for examination. Only this much they have given. So with this particular concept is enough. You don't need to worry about all the other factors and how they are exactly activated. You don't need to worry about this. Just only these things you please remember, that is enough. Clear? Now let us see. And now in the class 12th chapter called Principles of Inheritance and Variation, we will study some genetic disorders. Due to some genes defects or gene mutations, we are having some diseases. Out of them, one important disease is hemophilia. Hemophilia. The gene for this hemophilia is present on X chromosome of human. Please remember this. In the genetics concepts, we will study this. Hemophilia. It is also called as royal disease because this disease was seen in the descendants of Queen Victoria. In the descendants of Queen Victoria, that's why it is called as royal disease. And it is also called as a bleeder's disease because in this particular case, what happens if there is a damage to the blood vessel wall? Now what happens? In the normal condition, blood clot should be formed. But in this case, in the case of hemophilia, what happens? One or more blood clotting factors are not present. Please remember, if blood clotting factors are not present, if they are not present, blood will not clot. If blood is not clotting, what happens? Even for small cuts also they will lose so much of blood. Even for small cuts also they will lose so much of a blood. That is the reason why loss, loss means what? Lack. Lack means absence. So absence of any of the blood clotting factors, absence of any of the blood clotting factors can result in this hemophilia. So that too, out of all these factors, what are the frequently observed conditions? Please remember, scientists have classified them again based on the frequently observed conditions in the human population. So in the human population, if we are generally observing, we are mainly observing three types of hemophilia. One is hemophilia A, other one is hemophilia B, and the third one is hemophilia C. And here, hemophilia A is due to absence of factor 8, factor number 8, and hemophilia B is due to absence of factor number 9, and hemophilia C is due to absence of factor number 11, not 10, 8, 9 and 11. These are the numbers. So please remember this. In this way, any blood clotting factor, if it is not present, that mechanism, that this particular pathway is there, no? This particular pathway, it will not be activated. So now this activation cannot occur. So blood clot cannot be formed. So in this way, blood clot is not formed. Now so much of blood will be lost. So that is the reason why now this is called as a bleeder's disease. So hemophilia is also called as a bleeder's disease. All these names were clearly given in class 12 principles of inheritance and variation. You should compulsorily remember this particular one. Clear? So in this way, hemophilia is one important example. Now I am giving you one sentence that is from class 12, chapter name is microbes in human welfare. What is the sentence they have given here? Streptokinase. Please remember these things. This is also from NCERT. Streptokinase produced by the bacterium that is called as Streptococcus. Bacterium Streptococcus is producing streptokinase, and we are getting that, and modified by genetic engineering. So using the genetic engineering, biotechnology, recombinant DNA technology, we are slightly changing this so that now this streptokinase is used as a clot buster. Clot buster. What is the meaning of clot buster? So you are breaking the clot. You are breaking the clot. Now you just imagine here. Why you want to break the clot? And where you want to break the clot? Please understand here. Suppose the clot is formed here. You don't want to break it. You don't want to break it. Then at which location you are trying to break these clots? Please remember, in some individuals, the flowing blood. Please remember, now I am talking about, now here regarding the clot buster, we are talking about flowing blood. Sometimes in some individuals, what happens is the flowing blood itself will become little bit thick. Slowly it will become thick. So that's why to those patients generally we will give blood thinners. Anyhow, these concepts are not there in NCERT. Just to make you understand about this, I am telling you these names. So we are giving some chemicals called blood thinners. If you are not giving blood thinners, what happens? Slowly it will become thick and it will become blood clot. So in this way, there are chances that in some individuals, due to various reasons, there are some chances that flowing blood may undergo clotting. If the flowing blood is undergoing clotting, that is dangerous. Please remember, if there is a damage at the blood vessel, clotting is occurring, that is good. But if the flowing blood is undergoing blood clotting, that is highly dangerous, because please remember, so from the heart actually the blood is starting here. So nearer to the heart, these vessels are having little bit more diameter. And when blood is keep on flowing here, slowly the diameter of the tube is keep on decreasing. And at the capillaries, it is the least. Yes or no? At the capillaries, the diameter is very less. Now you just imagine a big blood clot is formed and it is going and stopping somewhere at some capillaries. Suppose these blood clots are blocking some capillaries. Blood flow to that particular part will be stopped. Yes or no? Blood flow to that particular part will be stopped. Now slowly that organ will be affected. Suppose that particular organ is the heart itself. Please remember, from the heart also, even from that arch, systemic arch or aorta, so when the blood is coming out, don't think that it is not supplying to heart itself. There is a separate blood system or the supply system to heart also. And there also these arteries will be present, veins are present, and there also capillaries are present. Suppose some of these vessels are blocked because of the blood clot. Now what happens? Oxygen and nutrient supply to heart itself will be stopped. Then what happens? Heart function will be affected, and in this way it may lead to myocardial infarction. Please understand this. In this way, blood clots that are causing the blockage of blood vessels, so that it is leading to myocardial infarction, suppose, then heart function itself will be affected. Then what happens? It is highly dangerous. So in those cases, to break these blood clots, we are giving a particular chemical that is called as streptokinase, that is obtained from streptococcus and further modified by recombinant DNA technology, genetic engineering, biotechnology. Okay. So in this way, this particular one is used to dissolve the blood clots. It is used to dissolve the blood clots. Used to dissolve blood clots means what actually the blood clot is technically? It is having the fibrin proteins. Yes or no? Blood clot is actually having what? Fibrin proteins. So what this streptokinase is having? This fibrin lysins. Please understand these technical names. You may think that not given in NCERT, but there they have mentioned, right, in the class 12th chapter. You need to understand this. Blood clot is fibrin, and what this streptokinase is technically having? Fibrin lysine molecules. Yes or no? So that's why they are called fibrinolysins. So that's why this streptokinase is nothing but fibrin lysine. That is the reason why it can dissolve the blood clots. So in this way, what they are saying here, streptokinase produced by the bacterium streptococcus and modified by genetic engineering is used as a clot buster to remove clots from the blood vessels of patients who have undergone myocardial infarction that is leading to heart attack. This is what the information they are saying. So in this way, streptokinase is nothing but fibrinolysin clot buster. It is dissolving the blood clots. Clear? This is what another sentence related to again blood clotting only. You need to compulsorily interlink these things. Clear? Now let us quickly go with NCERT content. You know that when you cut your finger or hurt yourself, not just cutting finger, so somewhere suppose there is some cut is happening, then your wound does not continue to bleed for a longer time. It is not continuously bleeding for a longer time. After some time, it stops bleeding. What is happening there? That's what they are saying here. Usually, the blood stops flowing after some time. So, what is the average time we are saying? 8 minutes. Okay, 8 minutes we are saying. Do you know why? So, why this blood is not flowing after this? Blood exhibits coagulation or clotting in response to injury or trauma. If there is a damage that is happening to the blood vessels, now this blood will, to prevent the loss of blood, now it is showing coagulation. It is also called as clotting. So this is a response that is showing. This is a mechanism to prevent excessive loss of blood from the body. Loss of blood is called as a bleeding. So to prevent the loss of blood, to prevent the bleeding, this blood clotting is occurring. That's what they are saying. you would have observed a dark reddish brown scum that is formed at the site of cut or an injury over a period of the time. So now what happened? This blood clot, now you can see this as a dark reddish one. Yes or no? That's what they're saying. So dark reddish brown scum that is formed because of the blood clotting. That's what they're saying. Next one. It is a clot or coagulum. Okay, clot is also called as coagulum. Formed mainly it is having what? Network of threads called as the fibrins. Please understand this. What actually the blood clot is having? Fibrins. This is a network of some thread-like structures. Network of some thread-like structures, and these proteins are fibrin proteins, which in which dead and damaged formed elements. Formed elements means what? Cells of the blood are trapped. So actually this fibrin is forming the cross network. So damaged cells, now they are trapped in that. So because of this trapping, what happens? That gap will be completely sealed. That's what they are saying. So in this way they are trapped in this. Fibrins are formed by the conversion of inactive fibrinogen. Gen is inactive. Okay. So fibrinogens in the plasma by the enzyme called as thrombin. So how these fibrins are produced here? Fibrins are active form. They are produced from inactive form called as fibrinogens. And who is involving in this conversion? It is the thrombin. It is the thrombin, by the thrombin. Next one. How these thrombins are produced now? Thrombins in turn are formed from another inactive substance present in the plasma that is called as a prothrombin. And this thrombin is further produced from prothrombin. Prothrombin is inactive, thrombin is active. In this way it is produced from prothrombin. Next one. An enzyme complex. This is a complex. It is having different components in it. That is called as thrombokinase. You can also call this as prothrombinase. You can call it as no problem. So thrombokinase or prothrombinase is required for above reaction. That means for the conversion of prothrombin into thrombin, you need this thrombokinase or prothrombinase. Next one. That's it. That's what the under the reactions only they are mentioning only these reactions. Next one. This complex is formed by series of linked enzymatic reactions. Series of enzymatic reactions, that's why it is cascade process. But what are those involving? That process is not needed for you. Involving a number of factors present in the plasma in an inactive state. So all these factors generally they are present in inactive state. Only when there is a requirement, then only they are activated. So that now this cascade of process will occur. Sequence of process will occur. Okay, next one. An injury or trauma stimulates the platelets in the blood to release certain factors. So already when we studied about the cells, we studied about the platelets. Platelets are very important for blood clotting. Yes or no? So now here what happens? If the damage is from the internal side, now the platelets are releasing some factors. And these platelet factors, now they are helping in the activation of this particular mechanism. Suppose the damage is from the outside, tissue factors are released, and those tissue factors will help in the activation of this mechanism. So in this way, one is intrinsic pathway, one is extrinsic pathway. So in this way, platelets are releasing some components. They are involving in activation of these particular blood clotting factors. This is what they are saying here. Okay, next one. Factors which activate the mechanism of the coagulum. So in this way, platelets, they are ultimately involving in activation of this particular blood clotting mechanism. Next one. Certain factors released by the tissue at the site of injury can also initiate the coagulation. So in this way, in the intrinsic pathway, we are mainly talking about platelet factors. In the extrinsic pathway, mainly we are talking about tissue factors. So in this way, different factors are involving in the activation of this particular mechanism. Okay, in simple they are closing this particular mechanism. In higher classes, one after the other, step by step you will study all the things. Next one. Calcium ions play a very important role in the clotting. So they are very important. In almost all the steps you are observing presence of the calcium ion. If calcium ions are not present, then coagulation will not occur. Clear? With this NCERT content is completed. Now we will see some application type of questions here. So first thing is heparin. We are saying that heparin is anticoagulant. How this heparin is acting as anticoagulant? Which step actually it is controlling? It is actually controlling the prothrombin to thrombin step is there, no? That step is stopped by this particular heparin. So in the flowing blood, all these factors are present in the inactive form, right? So there prothrombin also there. So in the flowing blood, prothrombin should not be converted into the thrombin. So in this way, heparin is acting as anticoagulant. So heparin mainly targeting this particular activation of prothrombin into the thrombin. That step is controlled by this heparin. This is the major one. That is completed. Now the next example. There are some chemicals that are called as chelating agents, and these chelating agents generally they will bind with, generally they are binding with divalent cations and all. Sometimes they can bind with monovalents also, no problem. So what they are doing, so they are chelating with this, they are binding and interacting with them. So when these agents are binding with this calcium, then what happens? Calcium is not free for the reaction. If calcium is not free for the reaction, so those corresponding steps will be stopped. Yes or no? So in this way, here what we are saying, EDTA, sodium citrate, all these things, these two. So these are chelating agents. Suppose you are adding these agents to the blood, what happens? Suppose you have freshly collected the blood from an individual and then you are adding this particular one and mixing properly. Now what happens? Now these will bind with calcium. So in the blood, whatever the calcium is there, that entire calcium is attached to these only. Now calcium is not free for the reactions. If calcium is not present, now what happens? Reactions will not occur. So blood will not coagulate. This is what the important point is. So in this way also we can stop the blood clotting. So that's why generally when we are doing the research, so generally what we will do in the tube, first we will add EDTA, then we will collect blood from the individual, we will add mix properly. That's it. Now this you can store it for longer duration. So in this way generally we are storing the blood. Okay. Next one. Next one, there is an application question. Suppose you are keeping the blood in the refrigerated conditions at low temperatures. Now what happens? Please remember our temperature is 37 degrees centigrade, so that these enzymes are working normally, so that clotting is occurring. Suppose the temperature you are decreasing, you are not disturbing the remaining things, simply the temperature you are decreasing. Now what happens? The time it takes for the blood clotting will be further increased. Please understand, normal conditions it is taking 8 minutes. But if you are keeping this blood under the refrigerated conditions, then what happens? Blood clotting time will be increased. So now it will take so much of time to clot the blood. This is what the important information that you can link up with this blood clotting mechanism. Clear? With this, coagulation or the clotting of the blood concept is completed, and there are some other extra points are there, but I don't want to talk those extra points in this particular lecture. Regarding whatever the extra important points are there, for that I will make separate videos under the heading of extra lectures. So under those extra lectures you can go through that extra content. Okay, here I am mainly trying to convey you how easily, how exactly we are preparing for the NEET examination. Please remember, in the NEET examination they are asking only with respect to the NCERT content. So first thing is, you should understand each and every sentence and word of NCERT. If you are preparing that much, that is enough. But anyhow, sometimes what we do is, we will try to give some little bit extra information so that your concepts will become little bit more clear, so that you can easily prepare for the examination. Only for that purpose we are giving some extra information. But please remember, we are not doing MBBS in this class 11th and 12th. Please understand this. So that is the reason why you don't need to worry about each and every information regarding each and every concept. In that case, what happens? For single chapter itself, it will take 2-3 months. That is not needed. We are not studying like that. Whatever is our syllabus scope and limitations, as per that only we are studying. And that too what I am doing here. So in the regular lectures, so as per NCERT, we are covering the entire content. If some extra points are required, those points I am covering separately under extra lectures. So those who are interested in those extra points, you can go through those extra lectures also. Okay, this is what already I told about this particular plan of teaching. So you can go through those lectures also. Nice. Okay, with this particular lecture is completed. So we will meet in the next lecture. Thank you.